Aerobic respiration. Glycolysis is a series of chemical reactions in the cytoplasm of a cell which essentially splits glucose in two. In the process, a small amount of energy is released and captured in ATP. For many organisms, glycolysis is just the first step of three. First comes glycolysis, then the Krebs cycle, and then electron transport chain. Altogether, this is called cellular respiration. It is the process in which glucose is completely broken down and used to produce a much larger amount of ATP. Glycolysis has two distinct phases, an uphill preparatory phase and a downhill payoff phase. One way to understand the two phases of glycolysis is to compare the process to a downhill bike ride. Glucose has a lot of potential energy. A cyclist at an uphill starting spot also has a lot of potential energy. With little effort, the rider taps the potential energy and coasts to the downhill spot. But sometimes, to reach a downhill spot, the cyclist must first go uphill, in which the cyclist invests energy to get to the top of the hill. Glycolysis is similar. Although glucose is packed with energy, the energy is not readily released until energy from ATP is first added. When ATP is added to the reaction, the resulting sugar molecule now has two phosphate groups, which make it less stable than before, and ripe for chemical breakdown. In the payoff phase, three reactions now yield energy. In one reaction, each of the halves from glucose is attached to a high-energy phosphate group, and each donates high-energy electrons along with protons, which are also known as hydrogen ions. These are attached to electron carriers called NAD. The resulting NADH molecules are high-energy electron carriers that are used later in cellular respiration for an even greater energy payoff. The essential difference between NAD and NADH is that NADH carries one additional proton and two additional high-energy electrons. As glycolysis continues and more bonds from the two halves of glucose are broken, the energy released is quickly recaptured by attaching phosphate groups to molecules of ADP, creating energy-rich ATP molecules. Two molecules of water are also produced during glycolysis. Near the end of glycolysis, two ADP molecules take the remaining phosphate groups, forming two additional ATP molecules. By the end of glycolysis, a molecule of glucose has been broken down into two molecules of pyruvate. Glycolysis results in the production of two NADH and four ATP molecules. However, two ATP molecules were originally invested, represented by the two remaining ADP molecules. So, the net yield for glycolysis is 2 ATP per molecule of glucose, as well as 2 NADH. However, much more energy still remains in the pyruvate molecules. Cells get much more energy bang for their food buck if they now perform the Krebs cycle and the electron transport chain. The Krebs cycle picks up where glycolysis left off. Remember, in glycolysis, the glucose was split into two parts of pyruvate. Note that these two pyruvate molecules retain the original 6 carbon atoms from glucose, as well as most of glucose's energy. Before the Krebs cycle can begin, the pyruvate molecules must be transported from the cytoplasm, where glycolysis occurs, into the mitochondria, where the Krebs cycle takes place. To prepare for the Krebs cycle, pyruvate is modified in 3 quick steps involving NAD and another molecule called coenzyme A. In one of these modifications, carbon dioxide is released from pyruvate and eventually exhaled from the body. In the other modifications, the 2-carbon molecule that remains is attached to coenzyme A, while a proton, also called a hydrogen ion, and high-energy electrons are donated to NAD, yielding the high-energy electron carrier NADH. The new molecule is called acetyl-CoA. Because 2 pyruvate molecules emerged from glycolysis, 2 acetyl-CoA molecules and 2 NADH are now formed. Note that 2 of the 6 carbon atoms for glucose are now in the form of 2 carbon dioxide molecules. Acetyl-CoA enters the Krebs cycle where the remaining 4 carbon atoms that originated from glucose will soon be released as carbon dioxide. In the first step of this pathway, acetyl-CoA combines with water and a molecule called oxaloacetate. The first outcome of the Krebs cycle is the production of a molecule with 6 carbon atoms, 4 from oxaloacetate and 2 that originated from glucose. This 6-carbon molecule is rearranged by the removal and then the addition of water. Another important outcome of the Krebs cycle occurs next, when the molecule is stripped of 2 molecules of carbon dioxide, which are exhaled. At the same time, high-energy electrons and protons are donated to molecules of NAD+, creating NADH molecules. Another important outcome of the Krebs cycle occurs in the next reaction, which draws energy from the remaining molecule to join inorganic phosphate and ADP into a molecule of ATP. In addition to NAD+, another molecule called FAD plays a role as a high-energy electron carrier. The 4-carbon molecule donates protons and high-energy electrons to these carriers. The molecule then becomes slightly rearranged by the addition of water, after which more electrons and a proton join with NAD+ to form NADH. The final outcome of the Krebs cycle is the reformation of the cycle's starting molecule, oxaloacetate. The entire cycle occurs again with the second acetyl-CoA molecule. As the second round continues, more NADH, more carbon dioxide, more ATP, and more FADH2 molecules result. If these molecules are added to those made from the preparatory actions, we can see that all 6 of the carbon atoms from glucose have been released in the form of carbon dioxide. Although a small amount of ATP has been formed in the Krebs cycle, it is the NADH and the FADH2 molecules that represent the most energy of the cell. These electron carriers enter the next phase of cellular respiration, called the electron transport chain. NADH and FADH2 molecules must give up their extra electrons to the electron transport chain so that they may revert back to NAD+ and FAD and help in the Krebs cycle again. The electron transport chain uses the energy from the electrons to produce a large amount of ATP molecules. Note that the electron transport chain absolutely requires oxygen to operate. Because the Krebs cycle relies on a functioning electron transport chain to accept electrons from NADH and FADH2, the cycle will shut down along with the electron transport chain if oxygen disappears. The electron transport chain takes place in the mitochondria. This step is essential for producing enough ATP for animals and many other organisms to survive. The previous two steps of glycolysis and the Krebs cycle have produced a small amount of ATP from the breakdown of glucose. They have also produced NADH and FADH. Through the electron transport chain, the cell can now use the energy in NADH and FADH2 molecules to make many more molecules of ATP. The mitochondria has 2 membranes, an outer membrane and an inner membrane, and is essentially a bag within a bag. The proteins of the electron transport chain reside in the inner membrane. Both NADH and FADH2 donate electrons to that chain. We will use NADH as an example. As electrons move from one complex to another in the chain, they transfer some of their energy to proteins. The pump proteins, also known as hydrogen ions, across the membrane. With each transfer, the electrons lose energy, which is used to pump more protons. The process of electron transfer results in a difference in the concentration of protons on the 2 sides of the membrane. This concentration difference, called a gradient, is a form of potential energy. The oxygen that we breathe is essential for electron transport. Oxygen is exceptionally electron greedy and snatches electrons from the end of the electron transport chain. After grabbing electrons, it combines with hydrogen ions to form water. From the operation of the electron transport chain, a concentration gradient of protons forms across the membrane. Like water held behind a dam, given the opportunity, the protons will tend to flow across the membrane barrier. A complex in the membrane provides a passageway for the protons and uses the energy from the proton flow to power the production of ATP. The protons will tend to flow in this direction until the concentrations are the same on both sides of the membrane. That is, until the gradient disappears. If the electron transport chain stops for any reason, such as a lack of oxygen to capture electrons at the end of the chain, the gradient will quickly disappear, turning off ATP production. The NAD+ molecules left over from the electron transport chain are later recycled back to glycolysis and the Krebs cycle, where they will capture more high-energy electrons and form food molecules. ATP, produced in plenty during this last stage of cellular respiration, contains energy that had its origin in a molecule of glucose. Along the way, the energy took many forms, including the energy held by electron carriers, the energy released by transporting electrons in the electron transport chain, and the energy stored in a concentration gradient across a membrane. In the form of ATP, the energy originally in the bonds of glucose can be used to fuel cellular work.